Okay, so about fluorescence lifetime again of endogenous molecules now in live mouse cancer models. So I come from the same. Isn't it so loud? I don't want to disturb the other sessions going. I'm from the same lab from Keck Center, so we will see a similar images. Okay, so first the cancer model. So we are working with immunodeficient nude mice, and we inject human pancreatic cancer cells into the tissue of the mouse ear, which is very thin, very quite transparent, and also easy to image, easy to immobilize on a proper sample holder. About imaging, we are using, you have heard some details about the optical setup that we are using. It's a laser scanning microscopy, multiphoton, or second harmonic, or whatever you want to do. So what happens with when we use, what is this? At 740 nanometer excitation with our femtosecond pulsed laser, we can detect basically two-photon excited NADH. This is a emission spectrum of NADH, and we are detecting in this region from 450 to close to 500 nanometers. At the same time, what happens also is three-photon absorption, which works for exciting tryptophan to some extent. Of course, the probability is lower, but you have seen in the previous talk that it works. We detect the emission above 350. It's not really the peak of tryptophan emission, but we only use glass objectives, and those are not transparent below 350. And as you heard also with we have to use 890 nanometer excitation to excite with the two-photon absorption process FAD separately. So we can also observe FAD fluorescence in the kind of green-yellow spectral range. A bit about, you have heard a lot about fluorescent lifetime imaging, so just very, very briefly, we are using TCSPC, that means time-correlated single photon counting, where you count your individual photons, measure the time difference to the nearest pulse, and build a histogram. Of course, we have to do some sort of fitting to extract two exponentially decaying components. And you can see this is like the more or less almost the full model with all the three parameters, including some background and time shifts and impulse response function of your instrument. And also it's all these decays and all these fitted parameters, you have them in every pixels. We have 3 detectors, 2 excitations, so you have a lot of data to work with. Of course, we are not using all 6 combinations here. We only used the meaningful ones so far. So we have just basically only three channels to work with. Of course, when we do live imaging, there are also some images that are not directly related to FLIM. So this is an example of kind of not confocal imaging with a confocal microscope, where we observe the vasculature making shadows on the general autofluorescence of the mouse tissue. And this is very important for us because then we can really re-image the same spot on the mouse ear over and over again over quite long time periods. So this is just a detail of the vasculature in a healthy ear tissue. And for comparison, this is a tumor mouse. So the xenograft, the tumor has already grown to pretty big dimensions. This is just the top of the surface, and the tumor itself is about 2 millimeters big and 1.5 millimeter high. So after 10 days of of growth. So this is a little late for us for our 25x lens, which only captures a pretty small field of view. But still, we can again see the vasculature and the details to make sure we are looking at approximately the same spot on the mouse ear. Of course, other thing we can do with two-photon excitation or with the femtosecond excitation is second harmonic imaging. So this is just an example again. At the top of the dermis layer of the skin, we observe usually pretty strong SHG signal, and you can, because the signal is very strong, you can usually take very detailed images, and also this helps you identifying the proper spot. And also we have observed quite striking differences between the collagen structure between normal tissue and malignant tissue. So, but this is not directly related to the FLIM analysis we are most interested in. So the FLIM images come here. This is an example of NADH emission. We are the color scale is coding for tau m, which is the amplitude weighted mean lifetime. And you can see a lot of structure there in the skin. And also the images, they differ as you go deeper from the very surface. This is the epidermis, which is basically the layer of dead cells. And as you go deeper into the dermis, you can also see blood vessels, sebaceous glands. Fortunately, with this mouse, we don't have so many issues with hair and hair follicles. But and also we image typically up to depth of 250 or 300 microns, where already we still can observe a lot of intensity, but not really very fine structure anymore. Yeah, this is just an illustration how many layers of skins are there. It has very discrete horizontal structures, so that's why also the analysis of these images is a little more complicated than plain cultured cells. The other thing is that as we go deeper into the tissue, we increase laser power to get more or less the same photon counts, the same intensity of fluorescence at our detectors. So generally we start at something like 10 or 20 milliwatts, and as we go deeper, we have to increase the laser power quite a bit. But that at the same time means that we cannot use NADH to FAD intensity ratio because the intensities, of course, are proportional to square of the laser power we are using. But there are more reasons why this ratio is not really useful. It's differential absorption of the two laser wavelengths, and it's also other effects that make this ratio very, very non-reliable. Again, and the other thing with the skin, as I've mentioned, the strong structure of it, all parameters that we measure also depend on depth. This is an example of, again, FAD mean lifetime, and this is the tumor tissue, this is normal tissue, and this is like the surface, and here you go deeper up to 300 microns deep. So you can see that all the lifetime parameters are very different for the epidermis and for the dermis, and there is strong depth dependence. So when we go and combine more channels, what happens? This is an example of more or less comparable depth between a control mouse, this is a lifetime image of NADH and FAD at about 60 micron deep, and tumor mouse. We can see that in some places, like when you compare the main mass of the example, the lifetimes are shorter in the tumor case, but there is a lot of structure, very long lifetime, for example, from the sebaceous glands and other things. And what about tryptophan? It has been mentioned here a lot. So, well, unfortunately, in our case, we don't really see much of tryptophan. The signal that we are getting is extremely weak. What we only can see is that when we go to the top of dermis, we can see a lot of SHG from collagen, but that's basically all we can see, and above or below this layer, we basically cannot detect any tryptophan, so we can't really add to the discussion whether tryptophan is degraded in tumor or tryptophan is enhanced in tumor. Yeah, this is out of question here. You have heard about the ROI method we are using for processing our images. So this is just a sample of one of the images overlaid with ROIs that, of course, we have to exclude some structures of the skin. And because this gives you, as you have already seen in a host presentation, this gives you a lot of data, so you can construct histograms of, but this is not really very easy for non-trained eye. You can also extract some more information from this vast amount of data. For example, and as Horst has mentioned, our redox ratio that we focus on now is the ratio of bound fraction of NADH and bound fraction of FAD. So if you just plot the median values from the histograms, one thing you can see, of course, the depth dependence is there. This is the tumor mouse and this is the normal mouse. But you can also see quite significant differences between the two samples. If you look at histograms of this is data from 2 mice. The dark lines is tumor case, and the soft lines are the normal tissue. So even in the histograms, when you combine them in this way, you can see quite clear difference between normal and tumor tissue. Well, so that's more or less all we have done so far. So as a conclusion, we have working xenograft model. We can observe NADH and FAD fluorescence relatively deep into the mouse ear tissue. We have means to revisit the same spot on the ear over and over again. As I have not mentioned, the fluorescence intensity is not really what we can look at. So our parameter that is based on bound fractions of NADH and FAD is a suitable redox parameter to study here. What to do maybe in the future, because we really don't need the UV detector to detect the tryptophan because we can't see it, and it's not really very clever to use lifetime for detecting second harmonic because we can use any other detector for that. So maybe we can repurpose it to observe some other spectral range of NADH, and maybe this would help us improve some of the multi-exponential fitting routines. Also, what needs improvement is some methods of 3D segmentation to really deal with the layered structure of skin. And what we plan is to do finer time course to really observe the initial stages of growth of the tumor xenograft. Yeah, I can acknowledge David Katsathus for the injection in mice and funding from NIH. So yeah, thank you for attention.